Assalamu alaikum. Welcome to the first lecture of Bioinformatics. I think Enamul Hawk messaged me. Yes, auto entry still does not happen. It's because I have not sent invitations once again. I shall send it soon. I think a few more students will register for this course, maybe. So I will send it again next week. Then I think everyone can enter the class without the permission. Can everyone hear me clearly?
Yes, sir.
Loud and
Yes, sir.
clear.
Yes, sir.
Okay. In the last class, a CR was elected. So who is the CR? Is CR present?
No, sir. I guess he will come soon.
Okay, no problem. Let us start the lecture today. So it's an introductory lecture on molecular biology. So yesterday I already told that I would go through these terms because again and again we are going to face these molecular terms and we need to understand them. So we are going to study some biology today, actually, but from the light of CSE, obviously. So in this course, we are actually going to talk about three major things: DNA, RNA, and protein. So DNA, RNA, and protein are actually nothing but strings. We all know about strings, right? The string is an array of character. So we are going to actually look at an array of characters in this course and nothing else. To computer science students, DNA, RNA, and protein are nothing but an array of characters. So let us start with DNA. In DNA, there are four characters. Those are A, T, C, and G. These are the characters in DNA. A, T, C, G has its full form. A is adenine. Okay, I think the names are in the slide, so let me not write them again. Adenine, cytosine, guanine, and thymine. These four simple terms or simple basic elements actually make up the whole DNA. So are we clear on this fact? So if we understand this, DNA is actually two strings. DNA actually consists of two strings. So let me just put this into perspective through an image. We have all heard some one time or another that DNA is double-stranded DNA. So this is a double-stranded DNA. Let me just zoom in. This is, say, strand number 1, and this is strand number 2. But the funny thing is, we actually need only one strand. We don't need two strands, because if we know one strand, then we can automatically determine what the other strand is. If you look at the picture closely, the violet base always sticks with the green base. You can see the whole picture, you can check out. Here the green and violet are together, violet and green are together, violet and green are together. And similarly, the blue base is always stuck with the pink base. Here also blue and pink together, blue and pink, and so on. So these are called complementary bases. These are called complementary bases. So complementary bases, the complementary of base A is complementary of base T. That means A and T attached together. The complementary of base C is the complementary of base G. And so these two bases actually attach together. So suppose this is the first strand of the DNA. So we can automatically say that the second strand is, which is the alternate strand, that is AT, TA, CG, GC, AT, TA, because A's complementary base is T. We can already see it from here. Again AT. TA, because T's complementary is A. CG, C's complementary is G, and similarly G's complementary is C. That means if we know this one of the strands, then we can automatically determine what the other strand is using this complementary base pair formula. So can we understand this thing?
Yes, sir.
Sir,
Yes,
I have
sir.
a question, sir.
Yes, sure, go on.
Sir, in some cases where there are mutations, sir, sometimes the A doesn't go with T or C doesn't go with G. They interchange. So how is that handled in this case?
No, if the mutation occurs, it will actually occur in both the strands. I mean, you can't have C here and then T here. You cannot have that. Both will be manipulated at the same time. Okay?
Okay, sir. Okay, sir.
We shall go through mutation shortly. Don't worry about it. Mutation is very, very important. In fact, uh, the parts that we shall study after our mid exam will be totally based on mutation. Evolutionary algorithms are mostly based on mutation. So we shall go through those in detail. So let us put into perspective the length of the human genome or DNA. So DNA is in every kind of species, right? Or in dogs, cats, even in bacteria, there are DNA. But those DNAs are different from human genome or human DNA. So genome and DNA are actually used interchangeably. They are same things, nothing different. Human genome is actually 3 billion base pairs long. So what is 3 billion actually? 300 crores. So I would say that it is quite long. That means there are 300 crores of characters in a single human genome. Though we are very different from each other, our DNAs are more or less same. They are a bit different, obviously, but the difference is very, very small. Okay, so that's human genome for you, 300 crores long. So the question is, where is this genome? I mean, does this genome stay in our head or in our belly? Where does this stay? So this DNA is actually in every cell, in every chromosome. So a human body is made out of cells. We all know that. Cells means kosh in Bangla. So in every cell, there is a nucleus. In every nucleus, there are some chromosomes. In every nucleus, there are some chromosomes. In every chromosome, there is DNA. Have I made myself clear right here?
Yes, sir.
So there are many DNAs, but it is to remember that all DNAs are exactly the same. That means in my body, whether I am talking about the cell of my brain or if I am talking about the cell of my food, the DNA in all sorts of cells in all the chromosomes of my body are exactly the same. Is it clear?
Yes, sir.
But now the question is, if all DNAs are the same, how come our body parts are different? I mean, the scalp of your head or your brain cells or your blood cells, they are obviously very, very different from one another. The blood cells obviously have no relation, have no connection with the brain cells or the cells of your skin. They are quite different. So, what is the reason for this vast stern difference? The reason is the expression of DNA. So, what is this expression of DNA? Let us discuss. Suppose there are two brothers. Suppose there are two brothers. One brother is a great musician, and the other brother is doing drugs his whole life. Now, suppose both these brothers had the talent for music, but one brother nourished this talent, and the other brother nourished his talent for drugs only. That is why one of the brothers is now a successful musician, and the other one is doing drugs, staying in some drug addiction store. So similar case is for the cells. In some cells, one part of DNA is expressed and other parts are repressed. And in some cells, the other parts are expressed and one part is repressed. Let me draw this. Let me draw this. Suppose this is the DNA. Suppose this is the DNA of our skin cell. Suppose skin cell, or let's put things a little bit simpler. Tongue. It's a better example. Jibba. So what does tongue do? It mainly tastes food. That means it has the ability to taste what we are eating. So suppose in this DNA, there are many things like this part is responsible for thinking. This part is for hair growth. This part is suppose responsible for taste and sight. This is a tongue cell. Okay. Now for the tongue cell, which one of these places should be expressed?
Taste cell.
Taste. And all other places will be repressed. So this tongue cell will have the same DNA as the brain cell or hair cell. But unfortunately, the DNA inside this tongue cell will only express the taste property. The other properties will be repressed. Okay? Is it clear?
Yes, sir.
Now, to understand this more clearly, we will need the concept of genes. So I shall go through it shortly, but from a high-level overview, this is actually what is happening inside the body. Okay? Now, does anyone have any question up until so far? Okay, so let me proceed. So now let us come to the gene. Gene is actually these are small portions responsible for actually expressing different characteristics of the corresponding cell. These are actually all genes. So gene is basically a small portion of DNA, and there are thousands and thousands of genes in human body, human DNA, actually, human DNA. Small portion of DNA. Okay. So now let me go to a very important topic, that is central dogma. Before moving on to central dogma, I think I should introduce you to RNA and protein first. Then we should get into the central dogma theory. First of all, let us go to RNA. In order to move on to RNA, we shall need to understand two concepts. One is the concept of DNA replication, and next we shall go through the concept of transcription. So first of all, let us go through the concept of DNA replication and finish the basic concepts of DNA. So when we are babies, when we are a small baby, I'm sure that there were much less number of cells in our body than in our current state, because we have grown a lot from that time. So does anyone remember the term cell division?
Yes,
Cell
sir.
division.
Yes, sir.
Yes, I think we all remember this term, cell division. Okay. So what cell division does is it actually multiplies the number of our cells. That means suppose we had only 2 cells or 3 cells before, it's actually now creating more and more cells. We all know that. Now when we are creating or when we are doubling the number of our cells, we actually need to double the number of our DNAs also, right? Does everyone agree with this?
Yes, sir.
Yes, sir.
So there should be some kind of mechanism through which we can actually create more DNA. And I have already discussed that all DNAs of human body are the same. So these DNAs have to be the same. So the way new DNAs are created is known as replication. That means replication is the process through which one DNA is created from another DNA. Okay. So let us first see how DNA is copied. This DNA has to be the same as this DNA, right? We know that because all DNAs have to be the same in a single human body. So let's see how do this copy occur. This is a fairly complex picture, but it's actually very easy. Let me put it easily for you. Suppose this DNA, this was the original DNA, right? We want to create two more DNAs from this original DNA. What happens is that this DNA breaks up from the strand perspective. This DNA had two strands, and you can see that this strand has what? Broken up. So now there are plenty of single bases right here. These are all single bases. When a new cell is created, in that new cell, we have ATCG bases floating all around. ATCG bases look around for DNA single strands where they can sit with their complementary bases. So if this is A, this is A, T will actually join here. Similarly, if this is C, G will actually join here from our cells. These G's are floating actually. These free bases are floating in our cell. So they will try to join with single strand complementary single strand complementary bases. That is how when this double stranded DNA breaks up into two single strands, these free bases actually join onto those single strands and make up two separate DNAs, which are actually identical to the DNA that has been destroyed right now. Is that clear?
Yes.
So these two DNAs will be exactly the same as this original template DNA, because the single strand is remaining and complementary base pairs are going into the corresponding places. So the exact same copy is being made. So is the idea clear how exact same copy is made during DNA replication?
Yes, sir.
So DNA replicates, that's fine. But the thing is, our body is actually functioned through the use of protein. Protein is the most vital element of our body. So this protein is actually not the same as the protein nutrition. I mean, that is a different thing. This is a different kind of protein we are talking about. This is not those protein that we find in nutrition. I mean, I'm not talking about that. I'm not talking about protein starch here. I'm talking about genome protein right here. So I think we all heard about enzymes, right? Enzymes.
Yes, sir.
So what enzyme does is that enzyme actually facilitates all our activities of inside our body. Suppose one enzyme is responsible for breathing, one enzyme is responsible for controlling the oxygen level of our body. So different enzymes are actually performing different tasks. So enzyme is actually responsible for our physical activities inside our body, our biological activities. And enzyme is actually made from protein. And protein is made from RNA. And RNA is made from DNA. So to understand the functioning of our body, we actually need to understand how RNA is formed from DNA, and how protein is formed from RNA, and how enzyme is formed from protein. So it's a long thing, I think. But we actually don't need to remember the details here. The thing we need to remember is the basic concepts behind these things. Because when you are studying a journal, a medical journal, or a journal of any computational biology, these terms will actually come again and again. And even if you write any paper, you will have to put these things together in the paper because reviewers actually expect for you to write the technical terms from the biological perspective. So let us now understand first how RNA is actually formed from DNA and what is actually RNA. So suppose this is a portion of DNA. This is not the full DNA. I shall point to you that first. This is only a small portion of DNA. Now DNA is actually made up of two types of things. One is exon and one is intron. So what is exon and what is intron? Exon is something or some portion of DNA, the activity of which is known to us.
স্যার আপনার ভয়েস স্যার নেটওয়ার্ক সমস্যা হয়ে গেছে
স্যার আপনার কথা শুনতে পাচ্ছি না। হ্যালো।
But intron
স্যার আপনার নেটে সমস্যা মেবি
network connection is a bit problematic now. Let me just resolve it first. Okay, first tell me up to where I was clear.
Sir,
Did I reach this page?
Sir, you are talking about exon, the portion.
Exon. Okay, I started here, so this was not clear. I understand. Let me just check the internet connection first. 1 minute. Is my voice clear now?
Yes,
Yes, loud and clear.
Today, actually, I don't have an internet in my home, so I'm using mobile data. That's why I think some problem is occurring. Okay, let me start from here again. Restart. So this is actually not the full DNA. This is actually a small portion of DNA. Okay. Suppose this is some portion I. A DNA is actually separated into two types of regions. One is exon and the other is intron. We know the exact functioning of exon regions, but introns are mysterious to us. We do not know how introns work or activate. Now, RNA is a thing or is an entity which is actually made out of exons. Exons of a small portion of DNA, not the full DNA. I will again repeat. RNAs are made up of small portion of DNA and that too of only the exons. Is that clear? Hello?
Again, sir.
yes,
Can
sir.
you repeat it?
Yes, sure. RNAs are made out of exons of the DNA. But the thing to remember is that this portion is not the full DNA. It is only a small portion of DNA. So RNA is made out of exons of a very small portion of DNA. Is it clear now?
Yes, sir.
Yes, sir.
Now this RNA as mRNA. So what is mRNA? There are actually three types of RNAs. I shall go through them shortly.
Sir,
Let
M
me
for
just
messenger RNA.
M for messenger RNA. Now one thing to note is that this portion, is there a double strand here? or a single strand?
So single strand.
Single strand.
Single strand. RNA is always single stranded. Remember that. There is a picture of an RNA right here. Though it seems like it is double stranded, but truly it is single stranded because this portion has just coiled. I mean, suppose I have a long, I have a long, what should I say? I have a long strand right here, and what I'm doing is I'm actually just rotating this strand. So this may seem like it's maybe double or triple stranded, but it's actually not. It's actually coiled. So RNA stays in this coiled way, but it's actually single stranded. It's actually one full strand starting from here and ending here. Okay.
Sir, your voice is splitting up.
In RNA, there are no complementary. They are actually another thing to. Is it clear now?
Yes,
Yes,
sir.
sir.
Okay. So in RNA, there are actually four types of entities: A, C, G, and U. Do you remember what they were in DNA? In DNA, they were A, C, T, and G. The G and T. Same T.
Yes, yes, yes.
Okay. So right, it's a little bit different in RNA. It's a little bit different in RNA.
Sir, we can't hear you.
So we can't hear your voice.
Okay, can everyone hear me now?
Yes, sir.
Hopefully the internet will be better now. But if it doesn't, then I will have to take the rest of the class tomorrow in the next class. I have to cancel now. Okay, let's start. Okay, does everyone get the single standard part? Hello?
Yes, sir.
Okay. And is the screen visible? Okay. So now that we have understood the basic components of RNA and mRNA, so let's see the rest of the part. Okay, so we have already seen the construct of messenger
Sir, the
RNA.
screen.
The screen isn't visible?
No, sir.
No, sir.
Okay, let me share it again. Okay. Now that we have seen how to construct mRNA from DNA, let's see the other variants of RNA. So there are also tRNA and rRNA. I think students who had biology in intermediate already knows these things. tRNA means transfer RNA and rRNA means ribosomal RNA. Okay, transfer RNA and ribosomal RNA. And mRNA means messenger RNA. These are the three types of RNAs situated in our body. So is this clear to everyone? This fact?
I think you missed the mRNA the application part.
Okay. Let me just repeat this part in brief. Yes, mRNA is actually formed from the DNA part. So the DNA is actually very, very long. And this is only a small portion of DNA. So in every portion of DNA, there are actually two types of entities. Those are exons and introns. Exons are the parts of which we actually know what activity they are responsible for. But introns are mysterious to us, and we actually don't know how they actually, what they perform actually. We don't know that. So mRNA is actually formed from these exon parts combining together of a small portion of DNA. So is that construction clear?
Yes, sir.
Okay. Now tRNA and rRNA are a little bit different. tRNA and rRNAs are already exist in our cell. They don't need to be formed from the DNA. So these two types of RNAs are not actually dependent. These two types of RNAs are not actually dependent on our DNA. These are not dependent. They are already available in our cell by the creator. So actually, how does these three types of RNAs work together? Because we have already told in the other page that DNA is actually responsible for creating RNAs, and RNA is actually responsible. Sorry, and RNA is actually responsible for creating proteins. So by RNA, we actually mean all three types of RNAs. All three types of RNAs means mRNA, tRNA, and rRNA. These three together actually form the protein. One type of RNA cannot make the protein alone. You will need all three of these in order to make a protein to work. So let's see how this actually works. mRNA is actually the code for the protein. Okay, so what is the code? What does the code actually mean? That is the main question. So first, let me clear this page up and let me just draw some things. So suppose this is an mRNA. This is an mRNA. In this mRNA, there are obviously A, C, G, A, C, G, etc. Because we know that mRNA is consisted of A, C, G, and U. Okay. Now it is to remember that three continuous bases of an mRNA actually forms a codon. Okay. Three bases of an mRNA consist a what? Codon. This is one codon, and the next three forms another codon, and so on and so forth. So, do you understand the concept of codon? What does codon mean?
Yes, sir.
Yes, sir.
Codon is actually three bases of an mRNA. Okay. Now, okay, next let us go here. So, how many codons are there? How many possible codons are there? How many types of codons are possible?
Sir, 4 cube.
4 cube. It is because there are three placeholders, and in each placeholder, we can actually put in A, C, G, or U. That means we can put any of these four right here. We can put any of these four bases right here. We can put any of these four bases right here as well. That means this is 4 cube is equal to 64. That means 64 types of codons are actually possible. So this is actually a simple permutation combination problem. I won't go through the details here. I think everyone understands this part, that there are 64 types of codons possible, right?
Yes, sir.
Yes, sir.
Okay. From each of these codons, what is formed is known as amino acid. Amino acid. And a protein is actually made out of amino acids. Okay. So there are 64 types of codons. That means how many types of amino acids are possible? 64 types. 64 types. But in reality, there are only 20 types of amino acid. But we shall discuss this later on. So let us just assume. Let us just assume that there are 64 types of amino acid. For now, I shall discuss this later. How 20 types of amino acids actually come. So let me just assume that there are 64 types of amino acid for now. Okay. That means one codon is responsible for one amino acid. That means, suppose this is my mRNA. G, A, C, U, A, C, C. That means 1, 2, 3. Okay, one more. I will need 3. That means how many amino acids are there right here? 4 amino acids.
4.
These 4 amino acids join together and make a protein. Is it clear?
Yes, sir.
This is an mRNA. This is an mRNA, and this is actually protein. When these codons actually transform into amino acid and join together, then this becomes a protein. Is it clear?
Yes, sir.
Now the question is, we can understand that from DNA we can get mRNA, and from mRNA we can get protein. So what the hell does these two RNAs do? I mean, what is their function? We don't see them working anywhere. They have a very big role to play here. So let me show you what that is. So in mRNA, we can see that we have the codes. These are codes, right? These are one kind of code on what type of amino acid to produce. These are codes for producing amino acids. So these codes are actually carried by the tRNA. These codes are actually carried by the tRNA or transform RNA to the rRNA or ribosomal RNA. And ribosomal RNA is the one responsible for making that protein. Okay? That means this transformation does not actually happen automatically, like magic. It has to be transformed by someone. And that someone is the rRNA. That means ribosomal RNA. But ribosomal RNA is not the carrier. Someone has to carry this information to the ribosomal RNA. That carrier is the tRNA. Now, is this thing clear?
Yes, sir.
Yes, sir.
I think in biology we sort of memorized these things, but now we are actually understanding what is actually happening inside. Okay,
Yes, sir.
so this is how a protein is actually made up. So we shall see the details of proteins, but we have to wait for a bit. First of all, let me just introduce you how there are only 20 types of amino acids. Why not there are 64 types of amino acids? Let us actually see it first. So naturally, a protein is formed of amino acids. We can already see that. A protein is actually 200 to somewhat 500 amino acids long. That means there are hundreds of amino acids situated in a protein, and they are the ones which make up a protein. And I have already told you that there are 20 types of amino acid. There are 20 types of amino acid, not 64 types. So now let me explain why there are not 64 types of amino acids and there are only 20 types of amino acids. Let me explain that first. Okay. Let me first just show you one picture and you will understand this, I think. This is a table. This table actually shows which codons are actually responsible for the production of which amino acids. These two codons are responsible for producing what? This PHE amino acid. These codons are responsible for producing lysine amino acid. That means there are cases where more than one codons are responsible for producing only a single type of amino acid. Is that clear?
Yes, sir.
So what is this mapping known as? I mean, if I ask you this question, from amino acid, sorry, from codon to amino acid. If I want to know the name of this mapping, the mapping from codon to amino acid, what is this mapping called? Many to one, or one to one? What kind of mapping is this?
Sir, many to one.
Many to one. Many to one, right? Because there are codons which are responsible for only a single amino acid. That means many codons can be mapped to a single amino acid only. But the opposite is not true. One to many is not true because one codon is not responsible for the production of more than one amino acid.
Sir,
Yes.
is it always fixed for first to letter?
Okay, that's a very good question. I mean, in most of the cases, the last letter is only differing, right?
Yes, sir.
Yes, we shall actually see this in detail. Why this last letter only in mutation? In mutation, there are actually two types of mutations: synonymous and asynonymous. So while studying mutation later on, after the mid exam, we shall see the significance of why the last letter only differs. Okay,
Okay, sir.
but that's a very good observation, by the way. Yes. Any question? Any more question? Someone was telling something, I think.
So isn't it a three-dimensional array?
Analysis, but I did not understand the previous part. What kind of analysis?
Set a three-dimensional array na?
No, this is not a 3D array. I wouldn't say the 3D array. This is just a mapping shown here. I wouldn't say that it's a 3D array or something like that. Not that complicated thing. It is just one way of showing the mapping from the codon to amino acid. Just a fancy way, I think. It's a fancy way of showing a mapping. So the mapping is complete, and most of these things are complete. So now I think we are clear about DNA and RNA, but we are still uh, in need of knowing about the protein because we have not talked much about this entity. One more thing that I would like to explain here is alternative splicing. This term is actually very, very important. For some reason, I forgot to explain this part, but let me explain it now. So this was an oversimplified explanation of how mRNA is created from a DNA. So let us go into a little bit depth of how actually mRNA is created from the DNA. So this is suppose a small portion of DNA. So from this portion, since RNA is actually single-stranded, so this is a single strand. This is a single strand prepared for producing some mRNAs. Now, what are these? What do you think these are? These colorful portions? Are these exons or introns? Exons, right?
Exons.
So different combination of exons are actually going to produce different mRNAs. Okay? And these different mRNAs are further going to produce different what? Proteins. So this is a very simple concept, and its name is alternative splicing only because these exons are actually being put in different permutations in different places. That is why it's known as alternative splicing. Nothing fancy about this. So I wanted to discuss about this, but forgot. So I just read it right now. So now the final thing in this lecture is the protein. I did not talk much about protein. What is?
Sir,
Huh. I
sir,
can hear you.
does the creation of mRNA happen in the body, or is it an explicit thing?
It's obviously everything happens inside the body, but the difference between tRNA, RNA, and mRNA is this tRNA and RNA are already abundant in the cell itself, but mRNA is actually dependent on the DNA. This is the only difference.
Oh, I see. Thanks.
Okay. Okay. Now, protein has very much depth in its studies, and it is not possible for us to go into all of them. But I shall obviously go through the fundamentals which we require for reading papers or reading different journals. So, protein has actually four types of structures. They are primary, secondary, tertiary, and quaternary. These are the four types of structure of protein. Now, I will actually show you a very short video demonstration and explain every step from the YouTube, because that demonstration is actually very, very good, I think. And it will be, I think, beneficial for you to see what these proteins actually look like. Let me just go to the demonstration right now. Yes, it is already open right here. Okay. So, first of all, let us go through the primary structure, okay? Primary structure is actually the most simplest structure there is. Primary structure actually shows you what the protein is actually made up of, nothing else. So there you will see here there are 20 types of amino acid. All the names are here: alanine, arginine, asparagine, and I don't actually remember all of the names. I actually know a few of the names. Okay, and you don't have to actually memorize these things. There is no need to memorize. Just know that there are 20 types of amino acid. So let me just write something. Suppose let me just construct a simple string here. RNDC. RRDRNNCDDR. Okay, I think let us stop for a while because Adhan is actually going on. Let us start after 2 minutes after the Adhan finishes. Okay, let me start again. Ahmed, Sierra, are you there? Can you hear me?
Yes, sir.
Okay, Ahmed, kindly do one thing. I think you have already opened the Facebook group for us. Please provide the Facebook group link in the message section here, so that anyone who are new can join the Facebook group as well. Okay, thanks. Okay, now let us presume. What was I doing here? Yes. So suppose this is a string of amino acid. There are how many amino acids are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So there are 10 amino acids. So this is actually the primary structure of protein. I know it sounds really silly that this is the primary structure of protein. So what's so great about this? But primary structure actually denotes the simplest thing, which amino acids are in which order actually consist the protein. Okay, so this is the primary structure of the protein. So now everyone understand what the primary structure is.
Yes.
Okay. Now these amino acids do not actually float on cell plasma. They are actually attached with one another using peptide bond. So let us see how the peptide bonds look like here. Let me just, yes. So these are actually peptide bonds. Amino acid 1, amino acid 2, amino acid 3. These amino acids are joined by peptide bonds. Okay. Now we have, I think we have all heard about hydrogen bond. I do not expect anyone to know what actually hydrogen bond is, and there is no necessity of knowing so. But has everyone heard about hydrogen bond?
Yes, sir.
Yes, sir.
I think yes, we actually know about hydrogen bond. Now, between these chains, between these red mark chains, there are some hydrogen bonds possible. And depending on what type of hydrogen bond we are talking about, different types of chains and different structures of chains can be formed. So there are actually three types of such structures depending on hydrogen bonding, the way hydrogen bonds are formed. The name of those are, let me write them, alpha helix, beta sheet. This is beta actually. I mean, I've written it B because there is no beta in my keyboard. Beta sheet, or let me just write it like this. And finally, coil. Okay, let me just explain all of these one by one. Alpha helix is the most common, beta sheet is a bit rare, and coil is actually those structures which we know actually nothing about. We do not know much about coil structures. So let me just show you in picture right here. How does those things look like? So this is actually a alpha helix structure. So there are amino acid 1, amino acid 2, amino acid 3, amino acid 4, amino acid 5, amino acid 6, amino acid 7, amino acid 8, and so on and so forth. The amino acids are formed in a spiral way. This is the alpha helix secondary structure. Another type of hydrogen bond occurs when the structure is formed like this. This is beta sheet. Amino acid 1, amino acid 2, amino acid 3, amino acid 4, amino acid 5. Amino acids move on in this order. This is the beta sheet structure. And coil structure is something arbitrary, about which we do not know much about. Those we actually call the coil structure. So do we understand what the alpha helix, beta sheet, and coil structure is? And these are actually all secondary structures. There are actually three types of secondary structures in protein. Secondary structures. They are alpha helix, beta sheet, and coil. So is this clear? Hello.
Yes, sir.
So secondary structures are actually formed because of the difference in hydrogen bonding. Hydrogen bonding. Okay. Now the burning question is, does one protein only carry a single secondary structure. That means suppose I have a protein. Will this protein only carry the alpha helix structure, or can it carry alpha helix, beta sheet, both structures? This is the question. Is the question clear? First, tell me that. I will just rephrase the question. Let me just show you this. Suppose this is a protein. This is a protein.
Sir, can you please show beta sheet again, please?
Beta sheet. Okay, sure. This is the beta sheet.
It's not coiled, it's wavy.
Yes, beta sheet is not coiled. Coil is another structure. The third structure is coil. Beta sheet is different, coil is different.
Sorry, sir. I mean spiral. It's not spiral, it's wavy. Or
Yes. This is beta sheet. Now, my question was, suppose this is a protein consisting of only 10 amino acids, which is actually impractical, I know, but just for example, I am just showing. Now, can I say that this whole protein will remain in alpha helix structure or beta sheet structure or coil structure? Can I say this with guarantee that this whole protein will actually assume only one structure?
Sir, I think the three of the structures can be present.
Can be present, exactly. That means this part. Suppose this part can hold the alpha helix structure. Maybe this part can hold the beta sheet structure, and this part may hold the coil structure. Okay. That means protein amino acids can actually form into small groups of secondary structures. Is that clear?
Sir.
Now, a very, very interesting problem, which was a hot topic in early 2015, is the determination of secondary structure of protein. That means you will be given the primary structure of protein, and you will have to make prediction of which part of protein carries which type of secondary structure. You have to predict that using machine learning methods. Okay. So it was a very important problem by them, and I think still you have provision on working in such problems, because still models are developing and machine learning models are gradually getting better and better. So maybe if you work on some existing literature, you can make it better, you can develop it, and then you can publish papers in good venues. And the name of this secondary structure system is known as Q3 structure. Q3 structure. Why is this name Q3? Because there are three types of secondary structures. The latest trend after 2018 is the Q8 structure. So what does Q8 mean? Can anyone say what does Q8 mean? Hello?
There are 8 kinds of secondary structures.
8 types of secondary structures, yes. This coil part is actually divided into many, many parts, because currently the scientists have discovered more types of secondary structures, and so they are actually using the Q8 structure right now. So one of the recent papers of Q8 was published in the Bioinformatics journal in 2020, which is a very big name. So they actually worked on predicting the Q8 secondary structure in protein. So you can find relevant data sets in the internet. And the paper can be found also, I think, if you search. Okay. So this is actually all about secondary structure. So there are two more structures, but I shall not talk a lot about them. I shall explain very briefly the tertiary structures and the quaternary structures. Quaternary structures. The tertiary structures are mainly structures of protein that are represented in 3D. 3D structure of protein. Okay. So what is 3D structure of protein? Let me just tell you the detail. I mean, in 3D, these are secondary structures, right? These are secondary structures of protein the different portions of protein actually hold different spinary structures. So these secondary structure portions actually make some angles in between them and stay in 3D space coiled like that. So arrangement of protein in 3D space describing using angles, angular representation of the protein. Secondary structure is known as tertiary structure of protein. That means the hydrogen bond oriented representation of primary structure is the secondary structure. The angle oriented representation of secondary structure is the tertiary structure. Is it clear? So this is actually the tertiary structure of protein. So does anyone have any confusion right here? There is actually image of a tertiary structure right here, which is actually very complex to look at. This is a tertiary structure image. There are different secondary structures, and they are actually making some angles and placing themselves in the 3D. Okay. So any question up to here?
Sir, which book should we follow?
I don't think you should follow any book for this course. I mean, you don't need to actually. But for recommendation, I can actually put the name of one book in the syllabus. I uploaded it already. But I think class lecture should be enough. If you follow the class lecture, it should be good enough for you, I think. These lectures are mainly prepared from the master's course of BUET. I mean, I have taken some portions that are very relevant for undergrad course, and I have put up the course using the master's course taken in BUET of computational biology. So I don't think any other books should be needed because the explanations and video lectures should be self-explanatory.
Okay, sir. Thank you, sir.
Okay. The final structure is the quaternary structure. Now we all know that in a society where we live in, we cannot do anything alone. We actually need many people to make something or some effort fruitful. So protein is actually not very different from us. Proteins actually come together. Like this and form quaternary structures and function together. So this is actually quaternary structure. So there is nothing special about them. So you have to remember one thing, that proteins tertiary and quaternary structures are the ones which are stable. The rest two structures are not actually stable, and we cannot actually find them independently inside our cells. We can find these two inside our cells independently. These are stable structures. So that's, I think, all about protein. From the next class, we are going to start some new topics, which is actually more related to CSC, computer science. So I would recommend you to watch this video once before coming on to the next class. Actually, I still epigenetics and PTM, these top two topics are left actually, but they are very, very short topics. So I will cover them in the next class, in the start of the next class. So till then, I think today's class is actually over. And I think I will give attendance to everyone today. And also in the previous class, I will give attendance to everyone because some students are still registering. So I will take attendance from the next class, inshallah. Okay. So anyone have any question?
Sir, the Pfizer's vaccine, which we heard about, that it's made up of mRNA technology. So that
Yes.
means the vaccine carries some code, which you showed just right today. So it's just some code, and it gives to the tRNA, and then the tRNA then. I mean the process that you showed us.
Yes, your idea is
similar?
absolutely correct. For preparing vaccines, you actually need genome editing as well. So I will actually show you genome editing after the mid exam. So obviously you will need the RNA codes. Without RNA code, nothing will happen actually. No vaccine can come fruitful. So I think I shall end today's class here. So We'll see you in the next class.
Assalamu alaikum.